In this class, we will be dealing with the development of the kidney. So first a question to you. A 45-year-old woman was admitted to the hospital with complaints of dull and intermittent pain in both flanks and passage of blood clots in the urine. On examination, there was fullness in both flanks and the kidneys were enlarged, which was confirmed by doing a USG. IVP confirmed the diagnosis of bilateral polycystic kidney. So, the questions to you are: name the developmental sources of the permanent kidney. Give the embryological basis of multiple cyst formation in polycystic kidney. What is the embryological explanation of pelvic kidney? And what is the position of the suprarenal gland if the kidney is arrested in its ascent? So by the end of this class, you should be able to answer these questions. So a quick flashback, okay, before we go to the development, so that you are familiar with certain terms, and also it will help you to learn about the development of the entire urinary system. So this diagram represents the formation of the three germ layers. So you have the ectoderm, you have the endoderm, and you have the intraembryonic mesoderm. You can see the notochord, above that the neural groove, okay, which later becomes the neural tube. Now on either side of the notochord, the intraembryonic mesoderm is divided into three parts. What were they? They were the paraxial mesoderm, the intermediate mesoderm, and the lateral plate mesoderm. So this is the same thing, okay, which is just showing the derivatives of the intermediate mesoderm. So you can see the neural tube again, and on either side you can see the intraembryonic mesoderm, which is divided into three. So the dark red, okay, that is showing the paraxial mesoderm, and all of you know that it gives rise to the sclerotome, the myotome, and the dermatome. The lateral most is the lateral plate mesoderm, and that is giving rise to the splanchnic, somatic, and also some extra embryonic. Okay, now what is left? It is the intermediate mesoderm. Okay, all of you have identified the intermediate mesoderm. Now from the intermediate mesoderm, your entire genitourinary system arises. Alright, so your genital system and your urinary system are arising from the intermediate mesoderm. So once this intermediate mesoderm is formed, how is it arranged? Okay, so you know that after all the germ layers are formed, okay, folding occurs. After folding, the intermediate mesoderm, it forms a bulging on your dorsal body wall. Okay, and this bulging lies lateral to the dorsal mesentery of the gut or your dorsal aorta, and extends from the cervical region to the sacral region. So look at this diagram. Okay, you can see the dorsal aorta and the dorsal mesentery of the gut. On either side of that, can you see two yellow circles? Okay, that is your intermediate mesoderm. You will understand much better with this diagram. Okay, so from cervical to sacral region, this bulging is extending. So the middle bulging is your gut, okay, and then it is connected by the dorsal mesentery, and you can also see the dorsal aorta there. On either side of this, you can see two elevations running down. Okay, that is called as the urogenital ridge. That elevation is called as, that longitudinal elevation is called as the urogenital ridge. Now look at the second diagram. Can you see the second diagram? Also identify the same structures in B and in C. Okay, you can see the urogenital ridge in B. Okay, now in C, what is happening to this urogenital ridge? It is differentiating into two parts. The urogenital ridge differentiates into two parts, a medial part and a lateral part. The medial part, okay, it is giving rise to the genital system. So we call it as the genital ridge. And the lateral part is giving rise to the urinary system, and therefore it is called as the nephrogenic cord. Okay, now you must be familiar with this too before we proceed. Okay, so this picture is showing the hindgut. Okay, it is showing the lower part of the hindgut, and you can see one diverticulum there. That diverticulum is called as the allantois. When you learnt about the yolk sac and the connecting stalk, you must have read about the allantois. So this allantois is a diverticulum from the yolk sac. Now the part of the hindgut distal to the allantois is called as the cloaca. Now look at the second diagram. You can see one septum. This septum is arising between the angle, it is arising in the angle between the allantois and the hindgut. Okay, and this septum is growing downwards. So a septum that is arising between the angle between the allantois and the cloaca, it is growing downwards. Okay, and then it is dividing the cloaca into two parts completely, an anterior part and a posterior part. Okay, so the ventral part, okay, the part ventral to the urorectal septum is called as the primitive urogenital sinus, and the dorsal part is called as the primitive rectum. So this is important when you learn about the development of the rectum, when you learn about the urinary system and also the genital system. So you must remember this urorectal septum and how it is dividing the cloaca. So now we will come to the development of the kidney. Now the kidney develops in the 4th week. It starts developing in the 4th week of intrauterine life. Okay, and it is developing from the nephrogenic cord. Okay, now the nephrogenic cord is lined by the coelomic epithelium, which is the epithelial lining of the peritoneal cavity. Alright, so during development, the kidney passes through 3 stages of evolution. They are pronephros, mesonephros and metanephros. So can you see in this diagram? Okay, can you see one red box, one yellow box and one green box? The red box is labeling the pronephros. The yellow box is labeling the mesonephros, and the green box is developing the metanephros. So the nephrogenic cord is forming 3 successive kidneys: pronephros, mesonephros and metanephros, succeeding each other in time and space, so that the last one to develop, which will be the metanephros, it will be the one that is becoming the permanent kidney. So pronephros is developing in the cervical region, and if you look in lower animals, okay, this pronephros is seen to be is the kidney in the cyclostomes and certain types of fishes. Alright, the mesonephros is developing in the thoracolumbar region, and this is the kidney in the case of amphibians and most of the fishes. Metanephros is developing in the sacral region, and it is the kidney in man. So this is following, I hope you have all heard of this term. Ontogeny recapitulates or repeats phylogeny. Okay, right. So the evolutionary history of kidney repeats. Ontogeny repeats phylogeny, or ontogeny repeats or recapitulates phylogeny. You must have heard of it before. Okay. So what does this theory state? It states that the development of an organism, that is its ontogeny, expresses evolutionary history and all the intermediate forms of its ancestors, meaning its phylogeny. So, recapitulation means that the development of an embryo follows the evolutionary history of an organism. So, our evolutionary history from lower organism to a higher organism, okay, is mimicked in the development of the kidney. So, in lower animals you had the pronephros, then in little higher you had the meso, and then in the highest you had the meta. So in the development of our kidney, it is also following the same steps. We first develop the pronephros, then we say goodbye to it. We develop the mesonephros, we say goodbye to it, and then we end up with the meta. Okay, so let us look at each part. Okay, so the pronephros, it is forming at the beginning of the 4th week in the cervical region. It is non-functional and it completely regresses. It is functioning, as we earlier mentioned, in certain types of fishes and cyclostomes. So look at this diagram. See the red box. Okay. See the part it is labeling. It is marked in dotted lines, not colored. Okay. That is your pronephros. So it is the highest part. So it is in the cervical region. Okay. And you can see that the pronephros has completely degenerated. It is not functioning. It is not at all present. But look at the pronephric duct. Follow those dotted lines down. You can see the lower part, it is persisting, and it is going and ending in the cloaca, opening into the cloaca. So a part of the pronephric duct is persisting. Actually, this pronephric duct is taken up or annexed by the next part of the kidney, developing kidney, or your mesonephros. You will understand when we go to the next one. Okay, so that is about pronephros. Now we come to the mesonephros. So this is a little more lower in the thoracolumbar region. Now these are functioning for a small period of time. So they are the interim kidneys. And this, if you remember that diagram, which you will see it, this diagram, okay. So you can see the mesonephric part. All right. So you can see the mesonephros, and you can see a series of tubules, excretory tubules that are developing in the mesonephros, and these are draining into the mesonephric duct. So mesonephric tubules, the excretory tubules that are developing in the mesonephros and are draining into the mesonephric duct. I hope all of you have understood. So most of these tubules disappear, but some of them are modified. And do you know what it takes part in? It takes part in the formation of the vasa efferentia of the testis. Okay, so we'll go back. So the mesonephros is in the thoracolumbar region. These are the interim kidneys. And you have the mesonephric tubules which are opening into the mesonephric duct. The mesonephric tubules disappear, but some of them persist and forms the duct system of the testis, or the vasa efferentia of the testis. Now what happens? So that is about the mesonephric tubules or the excretory tubules. Okay. Now what about the mesonephric duct? The mesonephric duct is the one that is giving rise to the ureteric bud, which you will learn in the subsequent slides. Okay, this ureteric bud is also called as the metanephric diverticulum, and this grows towards the metanephric blastema. So look at this diagram once again. Look at the mesonephric duct. Okay, see the mesonephric duct going all the way down to the cloaca, right? Now, before it enters the cloaca, you can see one diverticulum, yellow colored duct, is arising from the mesonephric duct. That is called as the ureteric bud. And this ureteric bud is growing towards, can you see that purple mass there? That is called as the metanephros. So this ureteric bud is growing towards the metanephros. So ureteric bud. So look at that diagram once again. Okay. Now this is growing towards the metanephros. Okay. So the permanent kidney is forming from the metanephros, and this is occurring at the 3rd month of fetal life. And in which part? It is in the sacral region. So this ureteric bud will drain into the ureter. So look at the second diagram. You can see the ureteric bud, okay, and it is giving rise to the ureter. Then the ureter will dilate to form the renal pelvis. Then you will have the major calyces, then the minor calyces, then the collecting ducts, and from there into the collecting tubules. So now you must remember that the metanephros, okay, it is having it's giving rise to your permanent kidneys. So your permanent kidneys, you can see it is, can you see that yellow portion and that purple portion? So it is having two components, right? So you have a ureteric bud, that yellow portion, and the purple portion, that is the metanephros. Okay, so first we will look at the ureteric bud. So the ureteric bud is giving rise to the collecting system of the kidney. That is, it is the one that is collecting the urine. So that part of the kidney is formed from the ureteric bud. The ureteric bud arises from the mesonephric duct, and you can see that it is growing cranially behind the peritoneal cavity towards the metanephros. Okay, now once it reaches the metanephros will form a cap. Okay, it will form a cap around the ureteric bud. When this happens, the ureteric bud will become dilated like a funnel, okay, and forms the renal pelvis. See the second diagram. You can see the dilatation, that is called as the renal pelvis. Then the ureteric bud will divide, okay, it will divide dichotomously for 13 generations and forms the collecting system of the kidney. The pelvis will undergo repeated divisions, okay, and forms, as we have already said, the major calyx, minor calyx, the collecting ducts, and the collecting tubules. So this is how the pelvicalyceal system of the kidney is formed. So ureteric bud divides repeatedly. The first 3 generations of branches fuse to form the pelvis. Then you know it in order: major calyx, minor calyx, the collecting ducts, collecting tubules. Ultimately, you have about 1 to 3 million collecting tubules. So this is again the same thing that we have discussed. You can see the ureteric bud developing from the mesonephric duct going towards the metanephric blastema. And you can see that once it reaches there, it forms a cap. Okay, and then the ureteric bud will start dividing and it forms all the parts of the collecting system. Now, what about the metanephros? The metanephros, so we have only developed the collecting system, that is the part that is collecting the urine. So we should now develop the part that is producing the urine, that is the excretory system. So the excretory system arises from the metanephros. You remember that purple ball that we saw in the previous slides? That is the metanephros. So the excretory part of the kidney is developing from the metanephros. Okay, so look at this diagram. Okay, now in this diagram, the yellow portion is your derivatives of the ureteric bud. Alright, now look at the blue one. That is your metanephric cap, which we said earlier. Okay, now what happens when the cells of the metanephros, they form the metanephric blastema. When the cells of the metanephric blastema come in contact with each collecting tubule, it forms a cap that is called as the metanephric cap. So you can see the metanephric cap. Okay, now look at the second diagram. This metanephric cap will then be converted into a metanephric vesicle. Okay, now look at C and D diagrams. Look at the shape of the metanephric vesicle. It becomes pear-shaped, then it becomes S-shaped. Okay, this S-shaped part is called now as the primitive renal tubule. Okay, now you can see that the proximal part of this S-shaped tubule, the proximal end, it is in contact with the collecting tubule, but there is no opening at present. Okay, it is still closed. So your metanephros and your ureteric bud parts are not fused yet. They are still separate. There is still a barrier between the two. Now look at the distal end of the tubule, S-shaped tubule. Okay, CE. You can see that it is becoming the Bowman's capsule. And how is it becoming the Bowman's capsule? By becoming invaginated by a tuft of capillaries, okay, that form the renal glomerulus. These tuft of capillaries are also developing from the nephrogenic cord. Actually, it develops from the angioblastic tissue of the nephrogenic cord. Now, look at the last diagram, F. Okay, look at the tubule now. Look at the S-shaped tubule. It expands and elongates and forms all the other parts of the nephron. So you have the Bowman's capsule. Okay, you have the PCT, you have the loop of Henle, and you have the DCT. So it is the DCT that is now in connection with the collecting tubule, and ultimately the barrier between the two will disappear and both the lumens will communicate. So that is the development of the excretory part of the kidney. So this is just what we have mentioned. Alright, so growing tip of each branch of the ureteric bud, that is the ampulla, solid clumps of metanephric blastema around each ampulla, then it becomes a vesicle, then it becomes pear-shaped, then S-shaped. Okay, the distal end is invaginated by capillaries and forms the glomerulus. Alright. Now, reciprocal induction. So, you saw that there was the metanephros and there was the ureteric bud. They were separate. Then the ureteric bud grew towards the metanephros. And once the ureteric bud and the metanephros came in contact with each other, they started developing. So this is called as reciprocal induction. The branching of the ureteric bud or the metanephric diverticulum is dependent on induction by the metanephric mesoderm, and it is controlled by certain genes, especially the WNT2 gene. It plays an important role in renal morphogenesis. So, in short, the kidney develops from two sources. The excretory part is developing from the metanephros and the collecting part is developing from the ureteric bud. Once again, that same diagram. Okay, so you can see the metanephros and you can see the ureteric bud. Now, the kidney has developed and you know that the kidney is now in the sacral region. But from your knowledge of gross anatomy, you also know that the kidney is not present in the sacrum. It is present above. So, it is actually where? In the thoracolumbar region, right? So, how did it go from the sacrum to the lumbar region? So, that is called as the ascent of kidney. Just the opposite of descent of testis. Here you have ascent of kidney. The kidney rises up. Okay. So, initially it was lying in the sacral region. And because it is going up, its blood supply also will change. So initially in the sacral region, it was supplied by the lateral sacral artery, and later when it ascends up, it is supplied from the aorta. As you all know, it is supplied by the renal artery, which is a branch of the abdominal aorta. So why does the kidney ascend? Okay, so the reasons are, one is the differential growth of the body wall and the reduction of the fetal curvature. Then you all know that the pelvis is quite small and there are a lot of structures already present there. So there is a space crunch. So for that, to escape that, it moves up and also to get a better blood supply. Then the kidney, initially the hilum of the kidney faces anteriorly, but during its ascent it rotates 90 degrees medially so that the hilum will now face medially, which is how you saw it. The fetal kidney becomes functional at the end of the first trimester. So that means it will start producing by the end of the first trimester. And it will start producing, what does the kidney produce? Urine. So this urine will be passed into the amniotic cavity. So it will mix with the amniotic fluid and then it is swallowed by the fetus. So anything that is wrong with this procedure, okay, in this process will affect the amniotic fluid amount. So either the swallowing, something is wrong, or either the passage and formation of urine, something is wrong. Both will affect the amount of amniotic fluid. So now you know the normal anatomy. So now let us look at what all can go wrong in the development of the kidney. These are called as the developmental anomalies of the kidney. 1. The kidney may not at all develop. The kidney will be absent. That is called as agenesis of the kidney. So agenesis means what? The kidney is not developing. So what are the developmental contributions from? It is from the ureteric bud. Okay, so the ureteric bud has to go to the metanephros. Then only the whole process will start. So if the ureteric bud fails to develop on one side, the kidney will not develop on that side. Without the ureteric bud, the metanephric tissue will not become the metanephric blastema. And only if the metanephric blastema forms, the ureteric bud can start developing into all its branches. Reciprocal induction. Okay, so renal agenesis can either be bilateral or unilateral. Bilateral is relatively uncommon because it is not compatible with life. Okay, and any newborn which is born like this will immediately or shortly after birth will die unless you can find a or do a kidney transplant. Now, this so both the kidneys are not there. So that means what? There will be no production of urine. So if no production of urine means the amniotic fluid will be very less. So when the amniotic fluid is very less, that is called as polyhydramnios. Sorry, not polyhydramnios. Polyhydramnios is more fluid. It is oligohydramnios. So when there is less amount of amniotic fluid, that condition is called as oligohydramnios. So why do we need this amniotic fluid? You must have all read about it or must have already learnt it in the previous chapters. We need it so that there will be an adequate medium, the baby can move, there is space for the baby to grow. So when there is oligohydramnios, the uterine walls will press against the fetus and the fetus cannot grow properly. And this condition, it can cause a syndrome that is called as Potter's syndrome. Okay, I will tell about it later in the next slide. What about unilateral agenesis of kidney? Only one kidney. No problem. We can survive with one kidney. The surviving kidney or the kidney that is there will grow a bit larger to accommodate extra function. And this is more common in males. And it's usually asymptomatic. Okay, the patient will not have any complaints. And while you are doing investigations for other conditions, you might find it as an incidental finding. So this is Potter's syndrome. See the baby? Okay. The face, because the uterine wall is pressing against the baby, okay, the baby is all crumpled and wrinkly, okay, and its face is distorted, its limbs are distorted, its skin is wrinkled, okay. So that is called as Potter's syndrome in renal agenesis bilaterally because of oligohydramnios. So can you see in this diagram, you can see if you look very carefully, you can see a faint outline of the kidney. The dye is injected into the ureter. You can see the bladder below. Okay, you are injecting the dye into the ureter and then it is going and filling up the pelvic calyceal system. And that is happening only on the right side. Left side there is nothing. So the kidney is absent on that side. Then you can have multiple kidneys. Okay, multiple kidneys occur when when the ureteric bud divides early. So there will be multiple inductions, okay, of the metanephric blastema. Okay, so multiple kidneys can form. So you can see here, you can see two kidneys on the right side. Okay. Now pelvic kidney. So this happens when the kidneys fail to ascend. That is called as pelvic kidney. Okay, so why this kidney is not ascending or going up? One of the common causes is presence of a sickle shaped fold of peritoneum that contains the umbilical artery, okay, which is projecting from the lateral pelvic wall. It prevents it from going up. So you can see here. Can you see in A? Look at the A. Don't look at B. Okay, we'll discuss B later. A, you can see. Can you see all the structures here? Can you see the adrenal glands and the kidney? And you can see the right kidney is down. Okay, it is not gone up. It is in the pelvic region. Now, renal ectopia. So what does renal ectopia mean? It is lying somewhere else. Okay, and one of them is that it is lying on, both the kidneys are lying on the same side. Duplication of ureter. This you can will come in when you are learning the development of ureter. Okay, I just put this diagram here. You can see on the left side two ureters, okay, going, and they are not joining, and they are draining two parts of the kidneys. Right side is normal. Malrotated kidney. So we said that it was the hilum was first facing anterior and then it rotated and became medially. So here that rotation is not taking place. So you can see here the right side, the hilum is facing anteriorly. Left side it is facing medially. Horseshoe kidney. Okay. So look at that diagram. The horseshoe kidney, the lower poles of the kidneys are fused. The inferior poles are fused. Okay. Now, this kidney, just like your normal kidney, will start to ascend from the sacral to the thoracolumbar region. But when it reaches the origin of the inferior mesenteric artery, look at the diagram. The inferior mesenteric artery won't allow it to go up anymore. Okay, so this point they can ask you, which artery prevents the ascent of the horseshoe kidney? It is the inferior mesenteric artery. Okay, so you remember from the dorsal abdominal aorta through three ventral branches, celiac trunk, superior mesenteric and inferior mesenteric. So the first one from below that you will encounter is the inferior mesenteric artery. So this artery will not allow it to go further up. Now look at this kidney, pancake kidney. From horseshoe kidney, only the inferior poles were fused. In the pancake kidney, the whole kidneys, both kidneys are fused to form one generalized mass that is present in the midline. This is a lobulated kidney. Okay, so fetal kidneys are normally lobulated, and this lobulation in certain adults it may persist. Normally we don't see lobulations in adult kidney, but sometimes this lobulated pattern may persist in adults. Functionally, no problem. So just the shape is different, that's all. Look on the left side. Then you have the aberrant renal vessels or the supernumerary renal vessels. These are relatively common. Okay, so what happens, you know that it is first arising in the sacral region and then it is going up to the thoracolumbar region. So as it is going up, it is taking blood supply from region to region, and ultimately when it reaches its permanent position, it is supplied by the renal artery. So as it goes up, the old one degenerates and a new one comes. Then it goes up again, the old one degenerates, and so here what happens? They persist. The fetal renal arteries will persist. So what is the importance of this aberrant arteries? And it is one of the common abnormalities, developmental abnormalities. What is the clinical importance? The importance is that because they may cross the pelviureteric junction, a lot of arteries, right, entering the hilum now, okay, they may cross the pelviureteric junction between the renal pelvis and the ureter, okay, the dilated part and the narrow part. Okay, so you remember that. And it will compress that region and obstruct the outflow of urine and give rise to, or it leads to, hydronephrosis. Hydronephrosis I will mention in the subsequent slides. So you can see here, can you see a lot of arteries that are going? Okay, so if it is compressing the pelviureteric junction, it can cause obstruction. So this is hydronephrosis. So first one you can see normal kidney. Okay, now look at the second diagram. See the pelviureteric junction. Okay, there is a small obstruction there, a mild obstruction. So what happens? Urine is not able to flow out properly. So the upper part or the proximal part, you can see that it is a little dilated. when you compare it to the normal kidney. Then you can see the moderate and then you can see the severe. So you can see how the dilatation is proceeding. So the pelvis will dilate, okay, then the calyces will dilate, then the medulla is dilating. Okay, so that is a severe case. It will end up in renal failure. The kidneys will be enlarged. It will be painful, okay, especially when the patient is trying to void urine. These are the, can you see the kidneys? And you can identify the abdominal aorta. Can you see the vessels going from the abdominal aorta to the kidney? That is the renal artery. So now you are having extra arteries on the left side. Right side, not the left. Left is normal. Now we have one condition called as the congenital polycystic kidney. Now look at this. You remember, look at the diagram. So you have the ureteric bud, okay, derivatives, and you have the metanephric blastema derivatives. Okay, so we said that, see that blue proximal end of the blue and the collecting tubule. Okay, so first there is no communication between the two. Okay, then by the end of the development there will be connection between the derivatives of the two parts, the excretory part and the collecting part. So that is what is happening normally. Now what happens if this continuation is not established? If there is a failure to establish continuity between the nephron and the collecting tubule, okay, the collecting tubules will start to show cystic enlargement. Okay, see the second diagram. Okay, you can see cyst formation in the tubules. So this is related to a condition called as the congenital polycystic kidney disease. It is a hereditary disease and you must have heard about it when you learned genetics. It is an example of an autosomal dominant inheritance. It follows an autosomal dominant inheritance pattern. It is not, even though it is congenital, it is not usually detected at birth. Okay, it is detected in the second or third decades of life and it never manifests before the age of 30. So you can see the normal kidney and the x-axis is showing the age. Okay, and the y-axis is showing the kidney function. So you can see the kidney is progressively enlarging. Okay, and the cyst formation is becoming worse and worse, and by advanced stage the kidney function becomes very poor. Patients will come with loin pain. I hope all of you know what the loin is. Hematuria, infections, hypertension, uremia. Uremia means urea in the blood. Urea should not be in blood. Okay. And CT image will show multiple cysts in both kidneys. So this is a sample. Okay, this is a dissected specimen of a polycystic kidney disease. Can you see the cysts inside and outside on the surface? Crossed renal ectopia, which I have already mentioned earlier, where both the kidneys are located on one side. Okay, these are all the different types of kidneys that we discussed. To summarize, I hope this flowchart is clear. The kidney is developing, having two components, an excretory part and a collecting part. The excretory part is developing from the metanephros and the collecting part is developing from the ureteric bud. So you must remember that the metanephros is the intermediate mesoderm in the sacral region, while the ureteric bud is coming from the mesonephric duct. Okay, so the metanephros will give rise to the metanephric cap, then your vesicle, then pear-shaped, S-shaped, Okay, and then invagination at one end by capillary plexus forming the Bowman's capsule, and then all the other parts of the nephron, PCT, loop of Henle, DCT, all will form. What about the ureteric bud? It gives rise to the ureter, then then it divides and fuses to form the renal pelvis, then divides dichotomously to form the major calyx, minor calyx, collecting ducts, 13 generations. Okay, ultimately the collecting tubules. Then these two parts will establish continuity. Other developmental events that we discussed were the ascent of the kidney, the arterial supply, its rotation, and then we talked about the developmental anomalies. So let's come back to the first question. 45-year-old woman, intermittent pain in both flanks and passage of blood clots in the urine, that is hematuria. So you examined the patient and you saw that there was fullness in both the flanks and the kidneys were enlarged. Okay, and you confirmed this by asking the patient to do a USG. You did an IVP and this confirmed your diagnosis of bilateral polycystic kidney disease. So developmental sources of permanent kidney. So the excretory part and the collecting part, you must write metanephros and the ureteric bud. Embryological basis of multiple cyst formation in polycystic kidneys. So these two parts, they are not establishing continuity, and therefore in the ureteric part there is formation of cysts. Embryological explanation of pelvic kidney, there is no ascent. Okay, what is the position of the suprarenal gland if the kidney is arrested in its ascent? So in our whole lesson we didn't talk about the suprarenal gland, except I showed you one slide. Okay, one slide of the pelvic kidney where you saw that the suprarenal gland was in its position. So suprarenal gland development is not related to the development of the kidney. So wherever the kidney is, no matter where the kidney is, the suprarenal gland will always be where the suprarenal gland should be. That is all. Thank you.